It's my pleasure to introduce you to my colleague, Dr. Lewis Scott. He's the commercial lead in immunology. Lewis, the floor is yours. Thank you.
Hi, Ramia. Hi, Ramia. Ramia, thanks for that introduction. I already met you. Let's get started. It's my pleasure to be here this afternoon. My name's Lewis, and I'm going to give you a little whistle-stop tour of our work here at ImmuONE and how we use cell painting routinely in-house. And this is part of the ImmuONE BYTES webinar series, some little bite marks coming into my face here, like macrophages that we're going to talk about a little bit later on. So I'll give you a little overview of what we're going to be talking about today. So we're going to go through a few topics, starting with high content imaging. We're then going to move into the role of the macrophage, its morphology, and how that's used in tox mechanisms. We're also going to be looking at image analysis for inhalation safety profiling. We'll then move into phenotyping and how that's used for individual cell responses. And then we'll wrap it all up in some takeaways at the end. So we'll get stuck in. So high content imaging, it integrates the phenotype of individual cells. And this was a technology that was started to be pioneered in the 1990s, and we now use it in phenotypic safety assessment. So it does a number of things. It applies automated microscopy with fluorescence detection and multi-parameter algorithms to look at basically how cells interact in populations. So it's pretty cool. And we can take this a step further in the realm of phenomics. Now, phenomics sounds a lot more kind of complicated than it is. It's essentially phenotyping, from the Greek to shine or to show, looking at visible traits, and omics combined, so that we can start to interrogate how cells and biological systems behave at every level of their biological organization, all the way from the transcript down to protein and metabolites, by taking imaging and next-gen sequencing proteomics approaches and combining that all together. And today we're going to look at one particular type of phenomic approach, which is high content image analysis. Now, we do this in a manner which is called cell painting. Cell painting is an assay which uses a collection of fluorescent dyes, and all of which label distinct subcellular compartments like the nucleus, the endoplasmic reticulum, and all of these compartments are fluorescently labeled. And then we can use algorithms which measure multiple features to generate a phenotypic fingerprint before or after compound treatment. Now, then we take that fingerprint and we interrogate it further by machine learning models so that we can look at the mechanism, mode of action, and even things like toxicity as well. And there's loads of different consortia public data sets that are available. I can send links over for these at the end if you're interested in pursuing these and kind of delving into some of that in your own time. So the phenotyping in vitro has a lot of power, and it's a really beautiful technique, I think. I know. And we look at early morphology and biochemical changes, and we link this to the mechanism of toxicity, which can then be correlated to in vivo endpoints. So the morphology readouts give a lot of meaning and context to markers such as cytokines, which, if you're looking at them on your own, isn't going to tell you a whole lot about that, how that system behaves, if it is an adaptive response, if it is an adverse response. So it kind of puts some kind of meat to the bone of how the kind of immune response is changing there. We can delve into mechanism of action, so we can interrogate specific pathways by coupling efficacy markers. And like I mentioned, we can correlate some of the morphological findings to the in vivo data. And we'll now kind of move into the alveolar macrophage response. So the alveolar macrophage is the first line of defense against inhaled substances in the lower airway, and has roles in homeostasis, disease, and infection that's pretty well understood when they act as almost these guardians for toxic responses in the respiratory system. And the responses have applicability in different contexts, such as phospholipidosis, apoptosis, and inflammation. So we'll kind of go on to some more of those in a second. So the macrophages have distinct morphology, which is really, really cool. And I get fascinated by looking at kind of the macrophages and all their different vacuolation changes as well. But these change in response to different substances or pathway activation. And that if we combine them with other markers, we've got a really strong determinant of pathway activation or mechanisms of tox. And all of this, at the end of the day, is to identify if something is an adaptive response to a compound, or is an adverse response to a compound. And we can use all of this data, we can take all of these findings together, and we can apply them to different contexts. So we can apply it to different tissue resident macrophages and look at macrophages in different tissues and contexts, but actually it's applicable to everything. I say everything, every cell type, so epithelial cells, fibroblasts, other cells. We can apply the same methodology to all contexts and our learning there to support a wide range of inflammatory questions. And I'm going to give you a little bit of a background of where we started as a company and how we have developed our methodology over the years. So we were tasked to solve a problem, and that problem was foamy macrophages, the responses of how macrophages respond to inhaled compounds, and could we generate an in vitro assay that could predict and be a read across for some of those in vivo findings, which we did. We solved the problem. We didn't just make assays for the fun of it, although they are quite interesting assays. But yeah, it was all about solving a problem that the industry had. So we characterized what the baseline was, which we did in 2017. There's a really lovely paper there. Go home and read it. It's I can definitely put it in the chat after. And then we looked at that baseline. We looked at how that baseline changed over time, the reversibility of it, and then started to think about how to apply it to other immune inflammatory contexts like M1, M2 phenotyping, and then taking that assay and putting a load of safe, putting a load of unsafe compounds on there, seeing kind of what happened if it could predict safe versus adverse effects, which it could. And then finally, we have applied it to other industries, which also had similar problems. So not just the pharma and therapeutic sectors. And goes up to June 2023 here, but actually we're going beyond there. We're looking into the future, and some of the work that we're doing at the moment in the poorly soluble space and looking at macrophage particle overload, which will present at some conferences this year. Some blinded studies with some of the results from actually 10 years ago when we were first tasked this problem, and then finally applying some of our methodology to macrophages in different contexts, like the oral space. So I guess what I wanted to say following all of that kind of hard work over years and years was that we have finally developed a standardized methodology from it that we use routinely with plants, and that is in its most simple state, taking macrophages, exposing to compounds of interest, looking at staining with fluorescent dyes, and then conducting the high content image analysis. We have this assay, which is called MorphONE, which we use routinely, which takes a panel of 4 stains, and we get a wide range of different features out of it. And look, there are loads of functional endpoints that one could say, oh, well, why haven't you used that? What about this? Why didn't you use that one? That could have been better. And ultimately, we consider these endpoints because they are relevant to morphological assessment of macrophages. And morphological assessment was the focus of this work and the problem to begin with. The direct link with standard alveolar macrophage pathology. And those findings in vivo studies where there were coarse and fine vacuolation properties, changes in size, we wanted an assay that would recapitulate that. And so we've taken this assay and we have run a lot of stuff through it. So we have done a lot of benchmarking, and you might think, well, actually, well, how do you know if something's safe in this model? Well, we've taken a large library of compounds, everything from published safe inhaled compounds to known toxicants, phospholipidosis inducers, antibody drug conjugates, some really nasty bits and pieces as well. And we have a really strong baseline or strong understanding of what baseline looks like in these models, what a safe response looks like, so that we know that that fingerprint is robust, well characterized. We know what a healthy cell is, so we can measure any deviations from that. And we have used that in preclinical context as an in vitro screening tool. And then can compare the mechanisms versus kind of other kind of compounds on the market. So it's it's become quite a useful tool in house. And we have over the years understood that there is a lot of power in phenotyping individual cell responses. That's kind of all that kind of publication history on that big timeline that I showed you, kind of the main point of that, and really that was found in one of the earliest papers in 2017, is that every cell reacts differently to a substance. Every cell in a tissue is going to be different. Look at this kind of little image here with all these different colored cells. All of those cells are different. There's heterogeneity. And so all of them are going to respond differently to a substance. And that variation can hide some key details if you're only looking at average results. So let's take this. We take bulk analysis and we take the average data. We have no idea what that the cell fate decision was, and so forth, and therefore we have no idea how that population responds. And we only have that kind of that average data that's generated there. Whereas if we are undertaking single cell analysis, we can start to reveal some hidden mechanisms and responses that the bulk analysis might miss, and therefore give us a greater understanding of the heterogeneity of that population. And why, well, how we select the characteristics. So we, like I mentioned earlier, there's a lot of characteristics we could have chosen, but we didn't choose all of them. You could collect hundreds and you could get more data than you could ever know what to do with in your life, and be completely bogged down by it, and it will crash every time you open it. Or you can be a little bit more sensible about it and think, actually, what endpoints are we interested in? Which ones are of particular interest? Let's go back to the in vivo studies. What were the histologists interested in? What were the changes that caused concern? And actually, those changes which caused concern were changes in cell area, vacuolation, and macrophages. So therefore, it wouldn't be appropriate to pick hundreds of them. Let's go for the characteristics which are going to be much more pertinent. And that's where we have the real power of individual cell analysis. So we'll take 3 or 4 that we're actually concerned about, and we can split that into 3 levels to generate 81 different phenotypes. So 4 characteristics, 3 levels, equaling our 81 different phenotypes. And the power of this will be revealed in a second, but let's start to talk into some data. So when you're looking at average data alone, I'm going to show you an example of a really nasty toxicant, and to be perfectly honest, here doesn't do a lot, barely does anything. Well, maybe it's not quite true. There's a little dip off in mitochondrial activity with that known toxicant, maybe a slight change in membrane permeability. But with test items A and B, you'd look at that and think, actually, that's fine. There's not really much going on in terms of mitochondrial activity, membrane permeability. It looks pretty safe. But actually, our assay is actually going to delve into that a little bit more and start to unpick some of the really interesting individual cell adaptations which are occurring in those populations. So what they did, well, what the team did in the study at ImmuONE was to take that and look at individual cell analysis, look at those different phenotypes. And so they would take phenotypes that were over 1% of the population and analyze them in detail and look at the effects that impacted over 20% of cells. Therefore, we could look at mechanistic changes that the average data missed. So let's take this one, for example. This is a population of cells associated with elevated cell area, elevated number of vacuoles. And as we see here, compared to the control, the test item A has a 3-fold change. And that's after 48 hours. But what's happening in chronic exposure? All of that would have been missed if you were only looking at the average data, because you'd think test item A looked pretty safe. And what about this one here? Okay, this is phenotype associated with decreased mitochondrial activity, increased cell area. And we actually see here that compared to the control, that toxicant has a really kind of really lovely shape, maybe about a 5-6 fold change there in the cells in that population that are responding with that elevated phenotype. Again, would have been missed in the average cell data. And this one here is another really lovely example. Cell population associated with decreased mitochondrial activity, increased cell area, increased number of vacuoles, and what we see is the toxicant really increasing the number of cells in that population associated with that phenotype. So the power of it lies in the individual cell analysis. And I have been talking for about 15 minutes, so I will start to wrap things up now. So I guess if I was to kind of give 3 take-home messages, they would be the following: why high content and why phenotyping? Well, the morphology, all the readouts that I've been talking about today, they give meaning and context to markers like cytokines, which on their own aren't going to tell you a lot about adaptive versus adverse. And if you're looking at single cell characteristics, that's great if you want to look at acute severe cellular tox. But if you want to differentiate individual cell populations and look into the mechanism of response, you're going to need something a little bit more sophisticated. Alveolar macrophages, why bother with them? Well, they have very distinctive morphological changes. The changes link really nicely to when combined with other markers, they can be good good determinants of pathway mechanisms, and their morphological changes link really nicely in vitro to what we find in the in vivo settings. And ultimately, why this approach? It's been developed over 10 years. It's very well standardized, very reliable, and we have that extensive database now where we have a really good understanding of what safe looks like in our hands and ultimately what the deviations from that might look like. So again, thanks so much for following this story with me, being part of this journey with me this afternoon. I will see you on the next ImmuONE BYTES, 26th of March. Come along to that. It's going to be going off. I don't think I'll be presenting it. Another one of the team will, but I'm looking forward to seeing you there. Please come along. All the publications I've talked about as well, have a read of them, and especially the 2017 paper as a bit of a primer to get you going. And yeah, thanks for bearing with me.